We have these three zones in the airways. So the first zone is the conducting zone. Conducting zone, trachea, bronchi, bronchioles, up till the terminal bronchioles. Generation 0 to 14 or 15. Now the generation 15 is called as second zone, which is transitional zone. And the generation 16 to 23, it is called as the third zone, which is the respiratory zone. So 16 to 23rd generation is called as respiratory zone. So in conducting zone we have trachea, bronchi, bronchioles up till the terminal bronchioles. And in transitional zone we have transitional bronchioles, and in respiratory zone you have respiratory bronchioles, alveolar ducts and cells. So in conducting zone, this zone does not take part in respiration, and it is called as contributes to the anatomical dead space. Maximum whatever volume we have in the respiratory zone. So anatomical dead space, its volume is approximately 150 m. Another important feature which you should note down is that the bronchioles do not have cartilage. No cartilage in the bronchioles. You will have cartilage only in trachea, bronchi and bronchioles do not have cartilage. Okay. In lungs, the major site of resistance is the large central airways. They are major site of resistance, center of resistance. Airway resistance is maximum in larger airways, central airways, trachea and bronchi, and the bronchioles contribute less than 20%. But in case of disease state, these so in the airway system, the trachea divides into bronchus, then to lobar bronchus, then to segmental bronchus, and then to bronchioles. Okay. So why this division occurs? Why this division occurs? This division has a sole purpose so that there is a minimal contact. Suppose there is air, this is tissue, and this is blood vessel. So this ramification of the bronchodivascular system occurs to increase the area of contact between the air and the blood and decrease the contact of the tissue. So for better air exchange, you should have minimal obstruction in between. The thickness should be low so that a single cell, ये जो thickness है एक single cell में reduce हो जाती है और air and blood का exchange better होता है. So tracheobronchial system and vascular system का ramification इसीलिए होता है. So trachea divides into branches. Similarly, The pulmonary artery divides multiple times into capillary system until it drains into the pulmonary vein. And trachea divides into bronchi, bronchioles, alveolar ducts, alveolar sacs. Majority of the compartmental distribution of lung volume occurs in the respiratory zone. Respiratory zone generation 16 to 23, it covers the maximum volume of the air and maximum surface area, and the trachea and central airways, they are the conducting system, and they hold approximately 150 ml of air. This volume of air is enough for the trachea. सो दिस कंडक्टिंग एयरवेज इनकी मॉर्फोलॉजी क्या होती है? ये कंडक्टिंग एयरवेज जो है ये टुवर्ड्स द पेरिफेरी डिवाइड होते रहते हैं डाइकोटमी से. इरेगुलर डाइकोटमीज मींस डिवीजन होता रहता है. दिस इज द ट्रैकिया, दिस इज द मेनस्ट्रम ब्रोंकस राइट एंड लेफ्ट. इट डिवाइड्स इनटू लोबार ब्रोंकस and then into multiple segmental bronchus and then to bronchioles and alveolar ducts and sacs. So this division or this dichotomy is irregular. और जैसे जैसे ये ब्रांच छोटा है ब्रोंकाई एंड ब्रोंक्यूल्स में, स्ट्रक्चर ऑफ द एयरवे ग्रेजुअली चेंजेस. इसमें जो कॉमन स्ट्रक्चर है वो क्या होता है? जितनी भी सेंट्रल एयरवेज हैं इनका 3 लेयर्ड वॉल होती है. 3 लेयर्ड वॉल, जिसमें से कॉमन जो लेयर है ये है म्यूकोसा, मसल लेयर and connective tissue sheath. First one is the mucosa, second is the muscle layer, and third is the connective tissue sheath. So the conducting system from level 1 to 14, they have this mucosal wall is pseudostratified columnar epithelium. Pseudostratified columnar epithelium with goblet cells. As you proceed from zone 1 to 14 or 15 up till the respiratory bronchioles, the size of the pseudostratified ciliated epithelium decreases and the number of goblet cells also. Ciliated cells of the trachea are columnar, whereas of the respiratory bronchioles they are cuboidal in shape. Cuboidal in shape. Size of the cells decreases as we progress from zone 1 to zone 14 or 15, and the number of goblet cells also decrease towards the periphery. And in bronchioles, the goblet cells are replaced by club cells. Club cells are present in bronchioles. These are the features of the mucosal layer of the conducting zone. What is the function of these goblet cells? They secrete mucus. The mucus layer is secreted into the epithelial cells by the goblet cell. These cells secrete mucus. These goblet cells, they decrease in number as we proceed from the conducting zone to the respiratory zone. So these mucus cells are replaced by clara cells, and the small bronchioles जो होते हैं these are devoid of mucus cells because they have, as we proceed further in the respiratory zone, they have surfactants within them. So this is the cartilage layer. This green layer is the cartilage layer. This red one is the muscle layer, the second layer, and this third one is the columnar layer. Pseudostratified columnar layer. So these are the 3 layers that are present. Muscle layer. This is the mucosa, first one. Second is the muscle layer. Third is the fibrous cartilaginous layer. Trachea and bronchi cartilage is in form of incomplete rings. They are in form of incomplete rings. In trachea and bronchi, the cartilage is in form of incomplete rings. The rings in the trachea cover the ventral and the ventral aspect and the lateral aspect, and the dorsal wall contains strong layer of transverse smooth layer. This layer is the transverse smooth layer. Posteriorly or ventrally, dorsally or posteriorly, you will have the posterior smooth muscle layer and this incomplete cartilaginous ring in green color, C-shaped cartilage in trachea and bronchi. These are the incomplete rings. After the division of the second and third generation of bronchi, these cartilaginous rings these cartilaginous rings they are replaced by incomplete cartilages. Like few cartilage, you will have few semilunar sheets of cartilages beyond the second and third generation of bronchi. One feature is the conducting layer is surrounded by, besides the cartilage, they have the external loose connective tissue layer. Connective tissue layer, which is the external layer. This is the fibrous sheath and this is the loose connective tissue layer. Besides cartilage, this is the loose connective tissue sheath which is present and it is continuous with the connective tissue elements of the lung. So what is the significance of this loose connective tissue layer? Outer loose connective tissue layer, what is the significance? The bronchial vessels, which are a part of systemic circulation, run in these connective tissue layer. Bronchial vessels run in these loose connective tissue layer. So these are part of systemic circulation. So the bronchial wall is supplied by the bronchial arteries, but only 25% of bronchial arterial supply is drained through bronchial vein. Rest of it goes through the bronchial venous plexus and drain into the pulmonary vein. And this contributes small amount to right to left shunt. This loose connective tissue layer besides the bronchial vessel, it is a site of lymphatic and the nerve supply also. Nerves and the lymphatic also run in this loose connective tissue layer. Besides the bronchial vessels, you will have the bronchus is accompanied by a branch of pulmonary artery. This pulmonary artery, this is also encapsulated by bronchovascular sheath या फिर bronchovascular space. जब भी leukocyte infiltration or edema formation happens, it happens in this bronchovascular space. So, what is bronchioles? Bronchiole is an airway which is devoid of cartilage. Airway which is devoid of cartilage. There is no cartilage in bronchiole. Devoid of cartilage. And it has no seromucous gland, which means it has no goblet cells. These bronchioles are part of conducting system and have a diameter less than 1 millimeter or less than or equal to 1 millimeter or less in diameter. They are only difference is that they are supplied with the blood from pulmonary circulation rather than they are supplied from pulmonary circulation rather than from bronchial arteries. Pulmonary circulation means pulmonary artery and from pulmonary vein rather than from the bronchial arteries. Another feature is their wall is thin walled and they are molded into the surrounding parenchyma. Another important feature is these bronchioles, they do not contribute to flow resistance of the airway, as they have lesser diameter, less than 1 mm, and airflow in these vessels is laminar rather than turbulent. The mucosa is cuboidal, absent goblet cells, but they have detoxifying and secretory function. These bronchioles are the last conducting airways. So they receive lot amount of air. In bronchioles, club cells are present in place of goblet cells. There are club cells present, and most important feature of bronchiole is that smooth muscle layer is very well developed. Smooth muscle layer is relatively thick and it is capable of narrowing the airways. Capable of narrowing the airway. Next is the alveolar ducts and sacs. Alveolar ducts and sacs. Their wall is opening of the alveoli. Opening of the alveoli. And their wall is made up of squamous epithelial cells. After the respiratory bronchiole, three generations from the alveolar ducts and then two alveolar sacs. So we have this grape-like alveoli which open into the alveolar sacs and the ducts. They are polyhedral in shape with mouth opening into the alveolar sacs. They are like cells within a honeycomb. Two adjacent alveoli के बीच में जो wall होती है, it is constant. It is called as interalveolar septum. The common wall between two alveoli is called as interalveolar septum. This is called as interalveolar septum. They are polyhedral in shape and this is the alveolar septum. Sometimes these septum are interrupted by pores. So what are these pores? These pores are called as pores of Kohn. These pores are covered with surfactant. So these collagen and the fibrous components ये extend करते हैं from the vulva to the hilum. और इसका फंक्शन क्या है? ये फंक्शन इसका है Transmission of the pressures from the chest and diaphragm. Transmission of pressure from chest and diaphragm. Alveoli की जो mouth होती है, the mouth of alveoli, they have smooth muscle, which is alveolar sac. They have smooth muscle there. Entrance. So suppose these are two alveoli, alveoli 1 and alveoli 2, and we have between interalveolar septum. This is the interalveolar septum with capillary running between it. Okay, so we have the alveolar epithelial cells on the both sides of the alveoli. This is the epithelial cells of alveoli 1. This is epithelial cell of alveoli 2. And these are single layer thin. So if you magnify it, we have this capillary. This capillary is also single layered endothelium. If we draw, suppose this is the capillary layer between the epithelial. This is the epithelial layer, this is the epithelial layer, and this is the capillary endothelium. Suppose this is the capillary endothelium. Okay, this we have the interstitial space, or in the interstitial space. So you have this alveolar epithelial cell, the basement membrane of the, sorry, we have the capillary basement membrane, the basement membrane on this side, endothelium, and the epithelium of the alveoli. So this constitutes alveolo-capillary tissue. Alveolo-capillary tissue ACTB. Alveolo-capillary tissue barrier. Alveolo-capillary tissue barrier. Alveoli में जो ये epithelium है, ये draw it in green color, जो ये epithelium है, its thickness is 0.1 to 0.3 micron. जब हम ये लाइट माइक्रोस्कोप में देखते हैं 0.1 to 3 micron, and this alveoli, they are continuous. These alveolar epithelial cells, they are also two types. Alveolar epithelial cells are also two types, type 1 and type 2. Type 1 is called as type 1 pneumocyte, and type 2 is called as type 2 pneumocyte. These are squamous cells. These type 1 pneumocytes, they contribute to 92% to 96% of the total alveolar surface area. The type 1 cells are poor in organelles like mitochondria and endoplasmic reticulum are less. They have cytoplasmic extensions. These cells have multiple cytoplasmic extensions. और ये कैसे बनते हैं? They form due to the division of type 2 pneumocytes by proliferation and differentiation of type 2 pneumocytes. These type 2 pneumocytes are type 1 is squamous and type 2 are cuboidal. इनको हम ग्रैनुलर न्यूमोसाइट्स भी कहते हैं। ग्रैनुलर न्यूमोसाइट्स और ग्रेट alveolar cell, great alveolar cells. They are small than type 1. They have no cytoplasmic extension. They have no cytoplasmic extension and they cover 5% of the surface area. They are rich in mitochondria, endoplasmic reticulum, and Golgi bodies. Mitochondria, endoplasmic reticulum, Golgi bodies are more in number. That's why they are called as granular pneumos. Granular pneumo. They have this very unique feature of having lamellar bodies. Lamellar bodies are very important in type 2 pneumocytes because they are the storage sites of pulmonary surfactant. These cells can regenerate by proliferation and therefore they are called as epithelial regeneration cells, epithelial regenerator cells during injury or pathological condition, and they are called as defender of the alveolus, defender of the alveolus. We have this third pneumocyte cells which are present in alveoli called as brush cells. Sorry, brush cells. So what is this interstitium? Interstitium is the space between the alveoli. Suppose you have this alveoli, two adjacent alveoli you are having, and suppose this is the alveolar epithelial cell, this is the alveolar epithelial cell, and this is the basement membrane of the, sorry, you have this basement membrane of the alveoli. Okay, and suppose this is the capillary endothelial layer. This is the endothelial layer of the capillaries, and this layer, suppose this layer in orange, this is the endothelial layer of the, sorry, this is the basement membrane layer of the capillary system. So the space between the basal lamina of the alveolar epithelium and capillary basement membrane or basal lamina of the capillary endothelium and the alveolar epithelium. Space between is called as interstitium. So this space is interstitium. This space in green color is interstitium. Interstitium. Okay. So we are going to read about interstitium. So what is interstitium? Interstitium is space between the basal lamina of the alveoli and the basal lamina of the endothelium. Okay, suppose this is alveoli, this is the epithelium cell of the alveoli, and this is the basement membrane. Okay, suppose this is the basement membrane, and this is our capillary. This is capillary endothelial cell, and this is the basement membrane of the capillary endothelium or basal lamina. Okay, so the space between this is called as interstitium. Okay, this area between the basal lamina of the alveolar epithelium, and the basal lamina of the capillary endothelium is called as interstitium. What does interstitium constitute? Okay, so these components of the interstitium are connective tissue and interstitial fluid. Interstitial fluid. In the connective tissue, we have cells, fibers, amorphous substance, and gel matrix. In amorphous substance, you have proteoglycans. Okay, these proteoglycans, not prostaglandins, it's proteoglycans. Okay, this interstitium has thin areas and thick areas of interstitium. Thin areas and thick areas. What does this mean? In thin areas, the amount of interstitial tissue is very less. You will have less fibers, lesser cells, less amorphous, so that the basement membrane of the alveoli, and the basement membrane of the capillary endothelium is almost fused. Okay, so why this happens? So as to increase the air exchange. So this thin area of interstitium mainly occur in respiratory zones where respiration is priority, and the thick areas, interstitium fibers, are more in number. Thickness of interstitium is more. These thin areas are called as restricted areas because it has been reduced to a thin basement membrane and single layer epithelial and endothelial cells. Here you will have almost no fluid accumulation. Whereas in case of thick areas, in cases of pathological conditions like lung edema, accumulation of fluid will be there. These are called as unrestricted portion of the interstitium. Unrestricted portion of interstitium. These unrestricted portion are mainly present in septal areas. They are The lymphatics are absent in these, are absent in septum. So how do the drainage of fluid occur? Drainage occur by continuous absorption via lymphatic in subpleural space and peribronchial connective tissue. The interstitium has three zones: peripheral interstitium, peripheral interstitium, axial interstitium, central and bronchovascular, central and bronchovascular, and third is the parenchymal. So this peripheral is present in subpleural space and interlobular septa. Okay. So suppose this is our lung. So this peripheral interstitium is present in subpleural space and between the upper lobe, middle lobe, and suppose this is lower lobe, interlobular septa. This is the peripheral interstitium. Second is the central or bronchovascular, which comes in axial. Okay, so axial is the bronchovascular. This is the peribronchial bronchovascular interstitium. So this is axial interstitium. Axial interstitium, peribronchial and central. Axial interstitium. Now the third one is you will have parenchymal, which is between the inside the intralobular. This comes in the parenchymal. So our pulmonary blood vessels, suppose this is our tracheobronchial tree. This is trachea, this is right mainstem bronchus, this is left mainstem bronchus, divided into upper lobe, middle lobe, and the lower lobar bronchus. So what happens is these pulmonary blood vessels, they take ramification across the branches of the tracheobronchial tree. Okay, so these branches will divide according to the tracheobronchial divisions. Okay, so these tracheobronchial divisions happens similarly. These pulmonary artery ramify along with trachea and tracheobronchial tree. Whereas the pulmonary vein, they have independent branching pattern. These pulmonary veins, they receive blood from several. Single pulmonary vein receive blood from several acinar units. Several acinar units. So pulmonary artery divides into multiple branches. They ramify along with the tracheobronchial tree. Suppose these are the multiple branches of the pulmonary artery. At the end, the peripheral branches form the capillaries. Suppose this is the capillary system, and in the end, this is the capillary system, and in the end, they join to form the pulmonary vein. So, this single pulmonary vein drains from multiple acini. Okay.